The oxygen-hemoglobin dissociation curve, also called the oxyhemoglobin dissociation curve or oxygen dissociation curve, is a curve that plots the proportion of hemoglobin in its saturated form on the vertical axis against the prevailing oxygen tension on the horizontal axis. This curve is an important tool for understanding how our blood carries and releases oxygen. Specifically, the oxyhemoglobin dissociation curve relates oxygen saturation and partial pressure of oxygen in the blood, and is determined by what is called hemoglobin affinity for oxygen, that is, how readily hemoglobin acquires and releases oxygen molecules into the fluid that surrounds it. Background. Hemoglobin, a globular protein, is the primary vehicle for transporting oxygen in the blood. Oxygen is also carried dissolved in the blood's plasma, but to a much lesser degree. Hemoglobin is contained in erythrocytes, more commonly referred to as red blood cells. Under certain conditions, oxygen bound to the hemoglobin is released into the blood's plasma and absorbed into the tissues. Each hemoglobin molecule has the capacity to carry 4 oxygen molecules. How much of that capacity is filled by oxygen at any time is called the oxygen saturation. Expressed as a percentage, the oxygen saturation is the ratio of the amount of oxygen bound to the hemoglobin to the oxygen carrying capacity of the hemoglobin. The oxygen carrying capacity of hemoglobin is determined by the type of hemoglobin present in the blood. The amount of oxygen bound to the hemoglobin at any time is related, in large part, to the partial pressure of oxygen to which the hemoglobin is exposed. In the lungs, at the alveolar-capillary interface, the partial pressure of oxygen is typically high, and therefore the oxygen binds readily to hemoglobin that is present. As the blood circulates to other body tissue in which the partial pressure of oxygen is less, the hemoglobin releases the oxygen into the tissue because the hemoglobin cannot maintain its full bound capacity of oxygen in the presence of lower oxygen partial pressures. Sigmoidal shape. The curve is usually best described by a sigmoid plot, using by a formula of the kind. A hemoglobin molecule can bind up to 4 oxygen molecules in a reversible way. The shape of the curve results from the interaction of bound oxygen molecules with incoming molecules. The binding of the first molecule is difficult. However, this facilitates the binding of the second, third and fourth. This is due to the induced conformational change in the structure of the hemoglobin molecule induced by the binding of an oxygen molecule. In its most simple form, the oxyhemoglobin dissociation curve describes the relation between the partial pressure of oxygen and the oxygen saturation. Hemoglobin's affinity for oxygen increases as successive molecules of oxygen bind. More molecules bind as the oxygen partial pressure increases until the maximum amount that can be bound is reached. As this limit is approached, very little additional binding occurs and the curve levels out as the hemoglobin becomes saturated with oxygen. Hence the curve has a sigmoidal or S shape. At pressures above about 60 mm of mercury, the standard dissociation curve is relatively flat, which means that the oxygen content of the blood does not change significantly even with large increases in the oxygen partial pressure. To get more oxygen to the tissue would require blood transfusions to increase the hemoglobin count, or supplemental oxygen that would increase the oxygen dissolved in plasma. Although binding of oxygen to hemoglobin continues to some extent for pressures about 50 mm of mercury, as oxygen partial pressures decrease in this steep area of the curve, the oxygen is unloaded to peripheral tissue readily as the hemoglobin's affinity diminishes. The partial pressure of oxygen in the blood at which the hemoglobin is 50% saturated, typically about 26.6 mm of mercury for a healthy person, is known as the P50. The P50 is a conventional measure of hemoglobin affinity for oxygen. In the presence of disease or other conditions that change the hemoglobin's oxygen affinity and, consequently, shift the curve to the right or left, The P50 changes accordingly. An increased P50 indicates a rightward shift of the standard curve, which means that a larger partial pressure is necessary to maintain a 50% oxygen saturation. This indicates a decreased affinity. Conversely, a lower P50 indicates a leftward shift and a higher affinity. The plateau portion of the oxyhemoglobin dissociation curve is the range that exists at the pulmonary capillaries falls 50 mm of mercury. The steep portion of the oxyhemoglobin dissociation curve is the range that exists at the systemic capillaries can result in the release of large amounts of oxygen for the metabolically active cells. To see the relative affinities of each successive oxygen as you add oxygen to the hemoglobin from the curve, compare the relative increase/decrease in P needed for the corresponding increase/decrease in S. Factors that affect the standard dissociation curve. The strength with which oxygen binds to hemoglobin is affected by several factors. These factors shift or reshape the oxyhemoglobin dissociation curve. A rightward shift indicates that the hemoglobin understudy has a decreased affinity for oxygen. This makes it more difficult for hemoglobin to bind to oxygen, but it makes it easier for the hemoglobin to release oxygen bound to it. The effect of this rightward shift of the curve increases the partial pressure of oxygen in the tissues when it is most needed, such as during exercise or hemorrhagic shock. In contrast, the curve is shifted to the left by the opposite of these conditions. This leftward shift indicates that the hemoglobin understudy has an increased affinity for oxygen so that hemoglobin binds oxygen more easily, but unloads it more reluctantly. Left shift of the curve is a sign of hemoglobin's increased affinity for oxygen. Similarly, right shift shows decreased affinity, as would appear with an increase in body temperature, hydrogen ion, 2,3-diphosphoglycerate or carbon dioxide concentration. Note: left shift, high O2 affinity. Right shift, low O2 affinity. Fetal hemoglobin has higher O2 affinity than adult hemoglobin, primarily due to much reduced affinity to 2,3-diphosphoglycerate. The causes of shift to right can be remembered using the mnemonic cadet, face right, for CO2, acid, 2,3-DPG, exercise and temperature. Factors that move the oxygen dissociation curve to the right are those physiological states where tissues need more oxygen. For example, during exercise, muscles have a higher metabolic rate, and consequently need more oxygen, produce more carbon dioxide and lactic acid, and the temperature rises. A decrease in pH shifts the standard curve to the right, while an increase shifts it to the left. This occurs because at greater H+ ion concentration, various amino acid residues, such as histidine 164, exist predominantly in their protonated form, allowing them to form ion pairs that stabilize deoxyhemoglobin in the T state. The T state has a lower affinity for oxygen than the R state, so, with increased acidity, the hemoglobin binds less O2 for a given PO2. This is known as the Bohr effect. A reduction in the total binding capacity of hemoglobin to oxygen due to reduced pH is called the root effect. This is seen in bony fish. Carbon dioxide affects the curve in two ways. First, CO2 accumulation causes carbamino compounds to be generated through chemical interactions, which bind to hemoglobin forming carbaminohemoglobin, and second, it influences intracellular pH due to formation of bicarbonate ion. Formation of carbaminohemoglobin stabilizes T-state hemoglobin by formation of ion pairs. Only about 5 to 10% of the total CO2 content of blood is transported as carbamino compounds. whereas is transported as bicarbonate ions and a small amount is dissolved in the plasma. The formation of a bicarbonate ion will release a proton into the plasma, increasing acidity, which also shifts the curve to the right as discussed above. 2,3-DPG-2,3-bisphosphoglycerate or 2,3-DPG is an organophosphate, which is created in erythrocytes during glycolysis. The production of 2,3-DPG is likely an important adaptive mechanism, because the production increases for several conditions in the presence of diminished peripheral tissue O2 availability, such as hypoxemia, chronic lung disease, anemia, and congestive heart failure, among others. High levels of 2,3-DPG shift the curve to the right, while low levels of 2,3-DPG cause a leftward shift, seen in states such as septic shock and hypophosphatemia. In the absence of 2,3-DPG, hemoglobin's affinity for oxygen increases. 2,3-DPG is a heteroallosteric effector of hemoglobin, lowers hemoglobin's affinity for oxygen by binding preferentially to deoxyhemoglobin. An increased concentration of DPG in red blood cells favors formation of the T-low affinity state of hemoglobin, and so the oxygen binding curve will shift to the right. Temperature does not have such a dramatic effect compared to the previous factors, but hypothermia causes a rightward shift, while hypothermia causes a leftward shift. Increasing temperature will weaken and denature the bond between an oxygen and a hemoglobin, which in turn decreases the concentration of the oxyhemoglobin. Carbon monoxide hemoglobin binds with carbon monoxide 200 to 250 times more readily than with oxygen. The reaction, HbO2 plus COHbCO plus O2, completely dissociates the oxygen molecules for the more favorable carbon monoxide, yielding carboxyhemoglobin. Moreover, because of hemoglobin's affinity for carbon monoxide over oxygen, carbon monoxide is a highly successful competitor. Even at minuscule partial pressures, carbon monoxide can displace oxygen. When carbon monoxide binds to hemoglobin, it increases the strength of the hemoglobin-oxygen bond. This increased affinity for oxygen means that it cannot be given up to the tissues as readily and results in a shift of the oxygen dissociation curve to the left. With an increased level of carbon monoxide, a person can suffer from severe tissue hypoxia while maintaining a normal PO2 because carboxyhemoglobin does not release oxygen to the tissues. Effects of methemoglobinemia. Methemoglobinemia is a form of abnormal hemoglobin where ferrous, which is normally found in hemoglobin, is converted to the ferric state. This causes a leftward shift in the curve as methemoglobin does not unload O2 from Hb. However, methemoglobin has increased RBC affinity for cyanide, and is therefore useful in the treatment of cyanide poisoning. Nitrites can be used to oxidize hemoglobin to methemoglobin, which can then bind cyanide. This restores cytochrome oxidase's ability to function. Furthermore, thiosulfate can be given to bind cyanide, forming thiocyanate, which can be excreted by the kidneys. Fetal hemoglobin. Fetal hemoglobin is structurally different from normal adult hemoglobin, giving HbF a higher affinity for oxygen than HbA. HbF is composed of 2 alpha and 2 gamma chains, whereas HbA is composed of 2 alpha and 2 beta chains. The fetal dissociation curve is shifted to the left relative to the curve for the normal adult because of these structural differences. Typically, fetal arterial oxygen pressures are lower than adult arterial oxygen pressures. Hence higher affinity to bind oxygen is required at lower levels of partial pressure in the fetus to allow diffusion of oxygen across the placenta. At the placenta, there is a higher concentration of 2,3-DPG formed, and 2,3-DPG binds readily to beta chains rather than to alpha chains. As a result, 2,3-DPG binds more strongly to adult hemoglobin, causing HbA to release more oxygen for uptake by the fetus. whose HbF is unaffected by the 2,3-DPG. HbF then delivers that bound oxygen to tissues that have even lower partial pressures where it can be released. Bins are also an effect of the oxygen saturation flexing its muscles.